Hello everyone. We shall now look at the most dramatic phase in the life cycle of a plant. That is, when it starts flowering. Flowering marks the beginning of the reproductive phase of the plant, during which the shoot apical meristem or the axillary meristems are transformed into reproductive meristems. There are various external factors that play a role in deciding the time of flowering. In the next two modules, we shall study the role played by some of these factors in the induction of flowering in Arabidopsis. As we know from the earlier modules, this model plant is very amenable for studying the molecular pathways underlying a development process. In this module, we shall see the role played by the photoperiod or the duration of light in bringing about the initiation of flowering. The learning objectives of this module are firstly to see how the vegetative shoot apical meristem becomes an inflorescence meristem. Then we will try to understand how a plant measures the photoperiod and responds to it. And finally, we shall study the genes involved during the transition to flowering. In Arabidopsis, the transition to the reproduction phase is accompanied by bolting or the elongation of the internodes on the stem, which during the vegetative phase are highly condensed to form a rosette of leaves. The inflorescence in Arabidopsis is of raceme type, where the inflorescence axis is indeterminate and individual lateral flowers arise sequentially from an apical inflorescence meristem. Hence, like the shoot apical meristem, the inflorescence meristem remains in an undifferentiated state. The inflorescence meristem first produces branches from the axils of colline leaves, which are borne on the inflorescence axis. Then the inflorescence axis produces flowers. The flowers are not subtended by bracts in Arabidopsis, though bracteate flowers are observed in many other species. The flowers are determinate structures that give rise to floral organs, after which no further growth occurs because the floral meristem is used up in forming the female reproductive organ. During the induction of flowering, the shoot apical meristem is transformed into the inflorescence meristem, as said earlier. During this transformation, the central zone of the shoot apical meristem, which is mitotically almost silent, becomes mitotically active. Due to this mitotic activity in the central and peripheral zones, the inflorescence meristem becomes dome-shaped and shows a new zonation consisting of an outer meristematic mantle indicated by MM in the figure to the bottom right, surrounding the central zone shown as CC in the same figure. The tunica corpus organization of the shoot apical meristem is however retained in the inflorescence meristem, and the floral primordia arise by periclinal divisions in cells beneath the two-layered tunica. Flowering in the appropriate season of the year is important for reproductive success of the plant. Premature flowering leads to insufficient vegetative growth and the lack of sufficient resources to sustain the plant, while late flowering may lead to the dispersal of seeds in unfavorable environmental conditions. Now, a default program for flowering initiation exists in all plants, which is kept repressed during vegetative growth. This was discovered when the shoot apical meristem of some mutants of Arabidopsis produced flowers instead of leaves almost immediately after germination of the seeds. These mutants were called embryonic flower mutants and are shown in the figure below to the left. These mutants bypass the rosette growth stage and germinate into a short inflorescence with a terminal flower and a few axillary flowers that lack petals and may be sterile. Hence, expression of EMF genes seems to repress flowering and are required for vegetative development. EMF1 encodes a plant-specific nuclear protein, and EMF2 encodes a polycomb group PCG protein. In plants and animals, the polycomb group proteins form multimeric epigenetic silencer complexes called polycomb group repressor complexes, or the PRCs, that bring about methylation of lysine 27 of histone 3 in target genes, leading to chromatin contraction and gene silencing. The EMF1 and EMF2 genes bring about silencing of the flower development program by interacting with and repressing all known floral organ identity genes. These repressors require to be displaced by selective transcription factors for initiation of flowering. The transcription factors may be induced by light duration, that is the photoperiod, or by temperature variation, or the plant hormone gibberellin. In addition, there is an autonomous flowering promotion pathway in which flowering is initiated independent of day length by another group of transcription factors. Age of the plant is also important for determining the time of flowering initiation. We shall see how these regulatory pathways lead to the transition to flowering. In this module, we shall study the role of the duration of light or photoperiod in bringing about the transition to flowering. Arabidopsis is a long day plant, which means that it flowers when the duration of light in a day is more than a critical period. In short day plants like rice, flowering takes place when the daylight falls below a critical duration. The photoperiodic behavior of plants can be studied by subjecting them to short days and then giving them a short exposure to light in the middle of the dark period. This mimics the effect of long days. Hence, flowering in Arabidopsis is accelerated by night breaks of light, whereas flowering in rice, a short day plant, is inhibited by such night breaks. In day neutral plants, flowering takes place irrespective of the light period. Tomato is an example of a day neutral plant. So, how do plants measure the photoperiod? All organisms have an internal clock called the circadian clock, which is built of a set of cellular proteins whose amount oscillates within period of 24 hours. These oscillations are autonomously maintained and enable the organism to synchronize their biological rhythm with the Earth's rotation. In plants, the circadian clock consists of three components, namely the central oscillator, which measures time, the input signals like light or temperature, and an output which regulates the developmental or physiological process. We will see each of these components of the circadian clock. The core oscillator consists of various components, all of which are proteins that are transcribed in such a way that they show peak expression at different times of the day. This is how the plant senses the time of the day. Two MIB transcription factors, circadian clock associated 1 or CCA1 and late elongated hypocotyle or LHY genes show peak expression at dawn, that is early in the morning, and minimum expression at dusk, that is in the evening. After CCA1-LHY expression, a set of genes called the pseudo-response regulators or PRRs are expressed sequentially at various times during the day. These include PRR 9, 7, 5, and 1. PRR1 is also called timing of cab expression or TOC1 and is expressed towards the evening. The PRR family proteins are unique to plants and consist of an N-terminal domain called response regulator that can be autophosphorylated in response to a stimulus. Finally, a set of genes which code for the transcription factor Lux arrhythmo or Lux and two plant-specific proteins, early flowering 3 or ELF3 and early flowering 4 or ELF4, are expressed in the evening and form a transcriptional regulatory complex known as the evening complex or EC. A membrane-bound protein, gigantia or GI, is also a component of the clock that is expressed in the evening. Together, these components of the core oscillator form autoregulatory negative feedback loops, repressing each other in a cyclic manner during the 24-hour period. Hence, CCA and LHY repress the genes that regulate their own expression as well as the expression of PRR and the EC genes. CCA1-LHY transcription in turn is repressed by PRRs, forming another negative feedback loop. The expression of GI is negatively regulated by the evening complex TOC1 as well as the CCA1-LHY. Besides these negative feedback loops, there are clock components like MIB transcription factors, REVIL, consisting of RVE4, 6, and 8, that are expressed during midday, which activate expression of several clock genes, including TOC1, the PRRs, and the evening complex. To activate gene expression, RVAs form a complex with the night light inducible and clock regulated genes, that is the LNK1 and 2 genes, which associate with the promoters of TOC1 and the PRRs. Hence, a balance between the expression levels of the activating and repressing transcription factors is important in the regulation of the core oscillator. The core oscillator, therefore, serves as a clock or a time sensor for the 24-hour period. In the absence of any external signals, the clock shows a free running period of 24 hours, which is reset each day starting from dawn, considered as time zero. As mentioned earlier, the core oscillator or the clock shows a free running period of rhythmic expression of the clock components for a 24-hour period in the absence of any external signals. After exposing the clock to alternating light and dark periods, the core oscillator components retain the same rhythm even when placed under continuous light or continuous dark. This is called entrainment of the clock. The clock can be also entrained by environmental signals such as changes in the light duration or temperature. If the light duration changes, say by having a shorter light-dark cycle, the clock may show a change of phase in the circadian rhythm. The clock then resets to the 24-hour cycle eventually, but there will be a phase advancement as compared to the phase during the free running period. Similarly, if the light period in the 24-hour cycle is increased, the clock may show a lag in phase, after which the clock resets to a 24-hour cycle but with a phase delay. Hence, the core oscillator is entrained by the input signals that it receives. Now, the perception of light is carried out by photoreceptors, which synchronize the light-dark cycles with the core oscillator. Plants have several photoreceptors like phytochromes, which are red light receptors, and cryptochromes and cytoloop, which are blue light receptors. These photoreceptors are present in leaves, in the cytoplasm or the nucleus. Let us see how phytochromes perceive light and entrain the core oscillator to the photoperiod. Phytochromes are photoreceptors that perceive light in the red region around 600 to 700 nanometers. The chromophore of these phytochromes is called phytochromobilin, which is a linear tetrapyrrole compound. The C-terminal region of the phytochrome has nuclear localization and dimerization domains. The chromophore is a photoreversible isomer which exists in two forms. On absorption of red light of 660 nanometers, the phytochrome exists in the PFR form, and on absorption of far red light of 730 nanometers, it exists in the PR form. Phytochromes are activated under red light, which brings about their isomerization from an inactive PR form to the active PFR form. Phytochrome A is active under red light of low intensity, while phytochrome B is active under high intensity red light. In the presence of far red light, the phytochrome in the PFR form of phytochrome B is converted back to the PR form. Hence, phytochrome B is a light stable and a red light sensor. Phytochrome A is light labile, and the PFR form degrades rapidly instead of being converted back to the PR form. The active PFR form undergoes autophosphorylation due to the kinase activity present in the molecule and migrates to the nucleus from the cytoplasm. In the nucleus, phytochromes aggregate to form subnuclear photobodies where phytochromes and downstream signaling components are co-localized. In the nucleus, there is an E3 ubiquitin ligase called as constitutive photomorphogenic or COP1, which forms a complex with the protein suppressor of phytochrome A or SP. COP1 is known to ubiquitinate several core clock components, example ELF3, TOC1 or gigantia, and mark them for proteasomal degradation in the dark. In light, the activated phytochromes interact with the COP1 SPA complex and inhibit the E3 ligase activity of COP1. Light exposure also promotes the nuclear export of COP1, thus preventing it from interacting with SPA1. This leads to the expression of the positive regulators of light responses and the clock genes. Phytochrome inhibitory factors or PIFs are basic helix loop helix type of transcription factors which are inactivated by phosphorylation. Activated phytochromes directly interact with the PIFs or the phytochrome inhibitory factors, which are negative regulators of light signaling and trigger rapid phosphorylation, ubiquitination, and degradation of the PIFs. PIFs are repressors of the circadian clock genes CCA1 and LHY. Inactivation of COP1 and the PIFs repressors by phytochromes lead to resetting of the core oscillator through expression of these genes at the appropriate times of the day. Cryptochromes are blue light receptors. They are also present in Drosophila, mice, and even us, and play a role in light-dependent regulation of circadian clock genes in these organisms. Cryptochromes have a chromophore consisting of flavin adenine dinucleotide or FAD that is bound to the phytolyase homology region or PHR at the amino terminal. Energy from the blue wavelength of light is used to reduce FAD to FADH2, thereby activating the cryptochrome. The carboxy terminal, called the C-terminal extension domain, shows kinase activity and brings about autophosphorylation of the cryptochrome monomers, leading to their dimerization. The Cry1 protein is present in the cytoplasm and moves to the nucleus when activated by blue light, while Cry2 is constitutively present in the nucleus. In the nucleus, the cries associate with other accessory proteins to form nuclear bodies and bring about regulation of light-induced genes, including the clock genes. In the presence of blue light, phosphorylated cries directly interact with the E3 ubiquitin ligase constitutive photomorphogenesis 1, which we have seen in the case of phytochromes, and brings about its inactivation. This prevents the E3 ligase activity of COP1, allowing the expression of the clock component genes. The Zeit loop or the ZTL proteins are photoreceptors of blue light, which play a role in regulation of the circadian clock. Zeit loop has a blue light absorbing domain called the LOV domain, which is responsive to light, voltage, and oxygen. The domain is bound to the chromophore flavin mononucleotide or FMN, which is reduced on blue light exposure. ZTL also contains F-box and Kelch repeat domains. The Kelch repeat domains act as adapters to get appropriate substrates to the F-box domain, which is a component of an E3 ligase. The ZTL E3 ubiquitin ligase brings about the proteasome-dependent degradation of the clock protein TOC1 in the dark. Binding of another clock component, gigantia or GI, to ZTL takes place in blue light. This prevents degradation of TOC1 in light and maintains the clock period. So crosstalk between the far red and blue sensing pathways occurs by the interactions of phytochromes and cryptochromes and Zeit loop at the protein level. The PIFs and COP1 represent hubs at which the photoreceptors converge to modulate the core clock components and integrate signals arising from the multiple wavelengths present in white light. Resetting of the core oscillator brings about a change in the output rhythms of cyclic developmental or physiological processes such that they can synchronize with changes in daylight hours during the year. If a plant is grown under a specific day length that causes these clock output proteins to be expressed during daylight, then flowering is induced in the long day plant like Arabidopsis or repressed in the short day plants like rice. A key clock output gene is a zinc finger family transcription factor called Constans or CO, which is expressed in the phloem tissue of leaves. Constans integrates the phase of the core oscillator and the light signal. When the core oscillator is entrained by the light-dark cycle through inputs received from the photoreceptors, Constans mRNA levels peak during the night under short days, whereas under long days, Constans mRNA peaks before the end of the day. Flowering is promoted specifically under long days because only under these conditions is Constans highly expressed while it is still day. In addition, Constans protein accumulates to high levels and is stabilized by light when Constans transcription peaks before end of the day and then shows a decline during night and during most part of the day. Phytochrome A and cryptochrome 2 are involved in far red and blue light dependent stabilization of the Constans protein through light dependent inactivation of PIFs and COP1 as seen earlier, while the protein is degraded in the dark by COP1. During the day, phytochrome B brings about the high fluence red light dependent Constans protein degradation by activation of another E3 ubiquitin ligase called high expression of osmotically responsive gene S1 or HOS1. Thus, only when the clock-driven high levels of Constans expression in the evening coincide with light-induced stabilization of Constans protein under long day conditions, does Constans activity reach sufficiently high levels to activate flowering. Constans expression results in the induction of the flowering locus T gene or FT gene in leaf phloem companion cells. FT encodes a phosphatidyl ethanolamine binding protein which binds to phosphatidyl ethanolamine lipids and moves through the phloem sieve tubes. FT protein is a mobile signal that moves from leaves to the shoot apical meristem and transforms it to the inflorescence meristem. Now, FT was called Florigen earlier and its existence was known since 1936 through grafting experiments which revealed that a graft transmissible signal moves from photoperiod induced leaves of the stalk to the shoot apical meristem of the scion to induce flowering. The nature of this signal, however, was not known till about 30 years ago when FT was discovered. On moving to the shoot apical meristem, the FT interacts with the B-zip transcription factor flowering locus D or FD. The FT-FD dimer activates the floral integrator gene suppressor of overexpression of Constans or the SOC1 gene and the floral meristem gene apetala1 or the AP1 gene, both being Matchbox type of transcription factors. The floral integrator and the floral meristem gene in turn activate downstream genes to specify the formation of flowering meristem and organs which will be dealt with in a subsequent module. The core clock components as well as the Constans FT module are conserved between long day and short day plants. However, the molecular mechanisms leading to FT transcription and hence flowering initiation are different. Rice is a short day plant and shows similar regulatory mechanisms for the photoperiodic control of flowering. The rice homologue of Constans is called heading date 1 or HD1. The FT homologue in rice is called heading date 3A or HD3A, which promotes flowering under short day conditions. As in Arabidopsis, the FT proteins move from leaves to the shoot apical meristem to induce flowering. HD1 activates HD3A transcription under short day conditions but represses it under long day conditions. The accumulation of the PFR form of phytochrome B leads to an inhibition of the clock component ELF3 under long days, but not under short days. Inhibition of ELF3 allows the expression of repressors grain number, plant height and heading date or GHD7 and the response regulator PRR37. Binding of these repressors to HD1 prevents the activation of HD3A under long days. Now, in the absence of phytochrome B inhibition of ELF3 under short days, the GHD7 and PRR37 expression is inhibited and this brings about the activation of HD3A and subsequent flowering promotion. Besides HD3A, rice shows other promoters of flowering, namely early heading date 1 or EHD1 and rice FT or RFT. In addition, non-functional alleles of the inhibitors GHD7 and PRR37 have also been identified, and such genes are thought to play a role in the photoperiodic adaptation of rice to a wide range of ecological and climatic conditions which led to the expansion of its geographical distribution. To summarize, in this module, we have seen how plants measure time through the circadian clock mechanism and how the clock is entrained to the light duration of a day. Inputs to this clock are given by photoreceptors, which are active in red and blue light. Regulation of the clock genes by the photoreceptors lead to the expression of output genes like constans in a rhythmic manner. Constans then leads to the expression of Ft, which moves through the phloem to the shoot apical meristem to activate genes that play a role in inflorescence and flower induction. Besides photoperiodic control of flowering, several other input pathways also regulate flowering initiation. These will be dealt with in the next module. Thank you.